Hey, it's Professor Gould, and in this set of lectures, we're going to talk about evolution at the population level and how we measure that. So first up, how do we know whether evolution is happening? We look for a change in allele frequency. And yes, we did do this when we were talking about genetic drift in the last lecture. We're going to do it in more detail in these lectures. So basically, we're just looking at, if we look at the whole gene pool, which is all the genes in the population, how much of that gene pool is made up of each allele, and is that the same from one generation to the next? That's actually the definition of evolution. We can look at this and measure whether the ingredients of natural selection are present, and then figure out whether evolution is actually happening in a population. One note we need to remember that natural selection can only select from the genotypes that are already present in a population. Natural selection can't make new genotypes happen. New genotypes and new phenotypes are created by mutations to nuclear DNA in germ cells. Remember, it has to be in the cells that can get passed on to the next generation. That's where new mutations come from. And these happen randomly. That is another thing to remember. Mutations are random in their development. So there is a thinking, and sometimes we talk about mutations as given the environment, this new mutation arose, and that's backwards. Mutations arise randomly. They may be more advantageous in a given environment, but there's nothing about the environment that requires new mutations to appear or makes new mutations appear. And this was actually proved by a very elegant microbial experiment many decades ago, and I'll post a link to that to make that make sense. Okay, so selection selects, mutations create new alleles. All right, so how do we know whether evolution is happening? We can thank Godfrey, Hardy, and Wilhelm Weinberg for figuring out a mathematical theoretical framework for figuring out whether evolution is happening and what would make evolution happen. And we call this the Hardy-Weinberg Principle of Equilibrium, which says that a population's allele and genotype frequencies are stable unless there is an evolutionary force acting on the population. So if there's no selection happening, then the frequencies are going to stay the same. Well, what does that mean? So that means that there are no new mutations that would create a new allele that would make a new allele frequency. There's no gene flow from another population. So there's no new alleles coming in. There's no natural selection selecting for or against any genotype. And this includes that there has to be random mating in the population because there's no sexual selection happening. And this is this is the one that tells you that it's just never happening. You have to have an infinite population size because you have to have a population in which genetic drift isn't happening. All right, so Hardy-Weinberg is going to involve math. I apologize for this. I'm going to break it down. If the breakdown doesn't help you understand it, it's not going to be on the test. Using the Hardy-Weinberg equations is going to be on the test. Okay, I'm going to show you how they derive. I'm not going to test you on that. Okay, so Hardy-Weinberg says that we have two alleles. This is in its most basic form. Yes, we can expand it to 3, 4, 5, however many alleles we want, but we're only going to use 2. Okay, so let's assume we have 2 alleles. One is dominant and one is recessive. P is what we use for the frequency of one allele, meaning the percentage of the gene pool that is that one allele. Q is the frequency of the other allele. And so if you put those together, you add up how much of the gene pool is one, how much of the gene pool is the other, you get 1 or 100% of the gene pool. Okay, so if we have 2 alleles, A, big A is dominant, little a is recessive, 46% of the gene pool is big A equals 0.46, That means big A is 0.46, little a is 0.54. You add those two together, you get 1. Okay, so we have these. This is our most basic Hardy-Weinberg equation. And if you know the allele frequency of 1, you can then figure out the other by just subtracting the known one from 1. That gives you the other. All right. Now, imagine a population where we have a 2 allele trait with simple dominance. So we've got 3 possible genotypes. And you remember all this from when we were doing Punnett squares. The total alleles in the population is the number is what we call the gene pool, and that is the number of individuals times 2, because everybody has 2 alleles for this trait. We are not talking about sex-linked traits. We are not going to talk about sex-linked traits when we talk Hardy-Weinberg. We are just going to deal with autosomal traits, because sex-linked trait makes this too complicated. We're not doing that. Okay, so take the number of individuals, multiply times 2. That's how many alleles are in the population. The allele frequency is the portion of the gene pool that is that certain allele. Imagine this is the chance that if you randomly picked an allele out of the gene pool, you would pick that allele. Okay? So if the gene pool is 50 big A, 50 little a, the chance that you would pick a big A is 50%. All right, so the genotype frequency is the chance that you would randomly select the genotype, meaning 2 alleles. So it's the chance of one allele times the chance of the other allele. And the way you get there is just the multiply the allele frequencies of the 2 alleles. All right, so the homozygous dominant genotype frequency is the frequency of the dominant allele times itself, so squared. Okay? Now, let's say that we have P and Q, right? P is the dominant allele, Q is the recessive allele. So to get the genotype frequency, or the percentage of the population that would have this homozygous dominant genotype, you just take P and square it. P squared, blah, blah. Okay. Q squared is the homozygous recessive phenotype. Q squared. Now, okay, this one is more complicated because probability is a bit of a bitch. Okay. Remember, when we're doing Punnett squares, there are 2 ways to make a heterozygous individual. Yes? Okay. You get dominant from mom, recessive from dad, or dominant from dad, recessive from mom, right? So, in order to get the probability for the heterozygous genotype, you take the little b frequency and the big B frequency plus the little b frequency, big B frequency, because we have to have, I should actually reverse those, because you have to have the 2 ways that you could get that combination. So it's not just P times Q, it's 2 times P times Q. All right? Now, if you add all of these up together, then you get all of the genotypes in the population, and that equals 1. So P squared plus Q squared plus 2PQ equals 1. And this is where my head explodes a little bit, but hang with me, okay? Because we said that P and Q equals 1, but then we do all of this complicated stuff multiplying, and it still equals 1. This is where my brain is like, no, that's not how math works. But it is how math works, because P and Q are both fractions. So if you add up 2 fractions and they equal 1, and then you do all of this complicated multiplication, remember that when you multiply fractions, they get smaller. And so P squared plus Q squared plus 2PQ also equals 1. All right. This is important. In the next section, we are going to use these equations to figure things out about populations over time. Stay tuned. It's cool.